Hi guys. I hope you had a wonderful Christmas and are about to have a good New Year. Sorry it's taken me so long to get this posted, but it is a crazy time of the year. So, um, this lecture will cover um, the structure and function of the plasma membrane. And as you should know, the plasma membrane is the, at least the theory of the plasma membrane is known as the fluid mosaic model. And the reason is because um, the molecules in the membrane are fluid, they're moving. And you can think of the plasma membrane as sort of the consistency of olive oil. So it's very viscous, but it is completely liquid. And it's actually the cell cortex, which is this area right here underneath the plasma membrane, directly underneath it, inside of the cell, which is mostly composed of actin filaments, but it has some other proteins and cytoskeletal elements associated with it that really maintains the shape of the cell. Now in plants that's a little different because you have um, the cell wall that also maintains the shape of the cell. But in animals, especially where there are no cell walls, it's the cell cortex that's responsible for the shape, while the membrane itself is very fluid. Um, in terms of number of molecules, the phospholipids are by far the most numerous molecules, but they are about equal in terms of mass with the proteins that are embedded within the membrane. Um, the lipid bilayer is termed such because you have these two faces of the membrane. You have the cytosolic monolayer, which faces where the phospholipid heads of the phospholipids face the cytosol, and you have the non-cytosolic monolayer, where the phospholipid heads face the extracellular fluid or the extracellular matrix or the cell wall. So your membrane is asymmetric. Okay, you have the cytosolic monolayer, and those lipids and proteins are going to be closely associated with things like actin microfilaments, um, whereas the lipids on the non-cytosolic monolayer are going to be associated with many carbohydrate groups, which are abundant in the extracellular matrix. Many of these are going to be attached to proteins, making glycoproteins, or attached to the lipids themselves, making glycolipids. We'll talk about those later. All right, so the plasma membrane has many functions, but at its most basic function is the separation of the inside of the cell from the outside of the cell. And then there are many functions that go along with that separation. So anything that involves the interaction between outside of the cell with inside of the cell. So communication between other cells, transport of things into the cell and out of the cell. And then, in addition to these interactions between the inside of the cell and the outside of the cell, the other function of the membrane is that it acts as a scaffold. So, in the cell, in the cytosol of the cell, unless things are held in place by cytoskeletal elements, especially the microtubules, proteins in general are diffusing through the membrane or through the cytosol. So if the cell is going to hold specific proteins into specific regions, membranes are a good place for that to happen. And we see that in organelles. We see the photosynthetic machinery embedded in the chloroplast membranes. We see the respiratory machinery embedded in the mitochondrial membranes. And we see many proteins responsible for building oligosaccharides or polysaccharides embedded in the membrane of the Golgi apparatus. So we see these on the inside of the cell, but you also see it on the outside of the cell. So scaffolding for biochemical activities can often happen in the membrane. In bacteria, which don't have internal membranes, you see aerobic respiration happening at the plasma membrane itself. But this isn't specific just to the primary metabolic processes, but there's other molecules that can be either produced or modified within the plasma membrane itself. All right. Also, I didn't mention movement and growth. Of course, the plasma membrane is integral to the growth of the cell, right? Because if the cell is going to grow, then the plasma membrane has to get bigger. So you actually have to add more membrane in order for the cell to grow. In addition, many of the movements of the cell happen at or because of the plasma membrane itself. So, for instance, amoeboid movement, including the movement with pseudopodia or filopodia that you see in fibroblasts. These are all the cell cortex, the actin filaments, and the myosin fibrils, which are pushing the cell membrane out. That cell cortex is capable of moving the cell itself because it's responsible for the shape of the cell. Also, you have your cilia and your flagella, and all of those are actually right under the membrane. The membrane actually sheaths cilia and flagella, so those are internal structures. All right, so let's talk about the different types of membrane molecules. Now, I'm assuming you guys are already familiar with the basic biochemistry of a phospholipid. Up here in the upper left, you see phosphatidylcholine, and you can see the basic shape of a phospholipid where you have the phosphate head group, you have the glycerol backbone, and you have these two fatty acid tails. Now, these fatty acid tails in phospholipids, especially membrane phospholipids, they tend to have one fully saturated fatty acid and one monounsaturated fatty acid, and that helps to maintain the proper viscosity of the membrane. But changing the saturation in these tails is a powerful way to change the melting point of the membrane, or more importantly, the freezing point. But most of the time, this phosphate head group actually gets short shrift. Okay, everybody knows it's a hydrophilic phosphate head, but it's not just a phosphate, right? This green part here is the phosphate. Okay, that's a P, and then there's 4 O's. But almost all phospholipids don't end with the phosphate attached to that third hydroxyl of the glycerol. They have an additional head group, okay? And that's what defines the different types of phospholipids. So here we have phosphatidylcholine because attached to the phosphate you have a choline group, right? You also may see phosphatidyl ethanolamine, right? That would have an ethanol group. You might see phosphatidylserine or phosphatidylinositol is important in cell signaling, where you have an inositol attached to the phosphate group. There's almost always going to be a hydrophilic group attached to the phosphate. Okay, but so when you see the phosphate head, it's not just a phosphate. It's a phosphate with another hydrophilic molecule attached to it. Okay, in animal cells, phosphatidylcholine is the major, the major constituent of phospholipids in animal cell membranes. All right, so phospholipids are the most common lipid in the plasma membrane, but there's other types of lipids as well, such as glycolipids. Okay, glycolipids are not phospholipids. They consist of a sphingosine, which is this fatty acid, with this nitrogenous group that attaches to it. Okay, so here's a nitrogenous group. And then you have another fatty acid attached there. And then attached to this other fatty acid, so attached here, this R group is going to be a sugar. Okay, now here, this is galactocerebroside. So there you have the galactose. Okay, so this could be any of many different oligosaccharides or even simple sugars like this galactose. The non-cytosolic monolayer of the plasma membrane is rich in glycolipids because it's those oligosaccharides that they actually make almost a cellular language that other cells recognize. And so when you talk about self-recognition, cells use these oligosaccharides to recognize self, to recognize this cell comes from my body. This cell does not come from my body. That's why the oligosaccharides on the proteins of red blood cells, they recognize self. Or on, you have to suppress your immune system if you get an organ transplant because cells recognize that the cells in this organ aren't from your body. All right, so this sphingosine group here, when it's attached to the fatty acid as it is in a glycolipid, together that makes what's known as ceramide. Okay, ceramide. So this orange plus this green is the ceramide. Then attached to it will be your sugar group. Okay. Notice that even though this isn't a phospholipid, we still have the hydrophobic fatty acid tails with a hydrophilic head group. Okay, not only in the sugar, but also in the functional groups in the sphingosine or in the ceramide. So you have an OH here, and you have a carboxyl group here and an amine group here. And so again, this is going to fit really well into the lipid bilayer with the hydrophilic head and the hydrophobic tails. Even cholesterol, which is almost completely hydrophobic, has a small hydrophilic hydroxyl group that allows it to properly orient itself within the membrane. Cholesterol is found in animal cell plasma membranes, and they act to maintain a intermediate fluidity. Okay, a lot of people want to say that cholesterol makes the membrane more fluid, but that's not actually, well, it's only true in some situations, and we'll talk about cholesterol a little bit more later. All right, so one more thing to mention about membrane lipids is that different cell types and different species will have very different compositions of their membrane lipids. So if you look at chloroplasts, they actually have a lot of sphingolipids, unusually high proportion compared to phospholipids. Also, the cytosolic monolayer will have a different a different membrane lipid composition than will the cytosolic monolayer, where you have a lot of glycolipids in the non-cytosolic monolayer, and you have a lot of, for instance, phosphatidylinositol is a signaling molecule that you find almost exclusively in the cytosolic monolayer. All right, so because of this amphipathic nature, where you have the hydrophobic tails and the hydrophilic heads, forming a bilayer, which is completely sealed is the most energetically favorable, most thermodynamically favorable conformation that a lipid bilayer or these phospholipids, these amphipathic lipids, can assume. So it's a spontaneous process. No energy is needed to form a self-sealing configuration. And so this also allows the budding off of pieces of membranes and the fusion of those membranes with other membranes. Okay, so you can see here you have the Golgi and you get vesicles that bud off and then they can fuse to the endoplasmic reticulum or they can fuse to the plasma membrane and vice versa. All right, the membrane carbohydrates, we talked a little bit about these. Membrane carbohydrates can either be found as glycolipids, where you have the oligosaccharides attached to the lipid. They can be glycoproteins, where you have oligosaccharides attached to proteins. And you also have what are known as proteoglycans. Proteoglycans are when you have a short piece of protein, the protein acts mostly to anchor the carbohydrate to the plasma membrane. And you have a carbohydrate attached to it. Okay, so a glycoprotein is mostly protein with an oligosaccharide attached to it. And a proteoglycan is a small piece of protein with a large carbohydrate attached to it. Okay, so really the protein functions to hold the carbohydrate, the polysaccharide. All right. Carbohydrates are found almost exclusively on the non-cytosolic monolayer. There are very few carbohydrates attached to the cytosolic monolayer. You can also classify membrane proteins, and there's different ways to classify membrane proteins. Of course, you can classify them according to function, and we'll talk about different types of proteins functionally, like transporters or receptors. But you can also classify membrane proteins based on their position within the membrane. Okay? Integral membrane proteins are not easy to separate from the membrane. So you must do something to destroy the membrane in order to detach an integral protein from the membrane. Okay? Integral proteins often span the entire membrane, and you can call those transmembrane proteins. So a transmembrane protein has a non-cytosolic domain and a cytosolic domain and a membrane spanning domain. Okay? Usually those membrane spanning domains are alpha helices, but they might be something else. You can also have integral membrane proteins that only span one monolayer. They're embedded only within a single monolayer. I don't have a picture of that here, okay? We'll look at that again in a minute. The transmembrane proteins tend to be things that have something to do with interaction of the inside of the cell with the outside environment, such as receptors or transporters, electron transfer proteins where they're moving protons from one side of the membrane to another. All right, so I want you to go back to thinking about the basic biochemistry of these molecules. So what kind of molecules are on the inside of the membrane? And so what kind of R groups would amino acids within those membrane spanning domains need to possess? If they're going to interact with the hydrophobic region of the membrane, then they must also be hydrophobic, right? Because hydrophilic molecules repel hydrophobic molecules. All right, so let's look at those membrane spanning domains. Okay, so here's the membrane spanning domain of a protein, and I want you to notice that this is an alpha helix. And these little green dots are the R groups. So these R groups must be hydrophobic. Okay, so the amino acids there are hydrophobic amino acids so that they can interact with the hydrophobic fatty acid tails. And in fact, they're going to be repelled by the aqueous environment outside of the cell and the aqueous environment inside of the cell. So they're happy to stay embedded in the membrane, and there's nothing else that is holding them there except for hydrophobic interactions. Okay, remember, this is the picture of the alpha helix when you're looking down it. This is not a channel, okay? This hole here, nothing can get through that hole, okay? But these are the amino groups and the carboxylic groups of the backbone of the protein, and those are going to be nestled within that alpha helix, so they are not interacting with the hydrophobic environment of the membrane. Okay, they're kept within that, tightly held within that alpha helix, and it's only these R groups that are interacting with the environment. Okay. Right, here's another picture. I love this picture. This is showing you all these little red and white dots. Those are water molecules. So you can see here is the membrane itself, which is completely excluding water. And then here is a channel protein, a porin protein. Actually, this isn't porin because those are beta-pleated sheets. But here is a water channel where the water molecules are able to pass through the alpha helices that are embedded in this membrane. Okay, so I want you to look at this protein, or this picture, and notice this is one of those proteins that is an integral membrane protein, but it doesn't span, it's not a transmembrane protein. It's simply embedded in the cytosolic monolayer. Okay, here is a transmembrane protein with a single pass. Notice this transmembrane protein is linking to another cell's transmembrane protein and holding them together. Uh-oh, I have to plug in. And notice that this channel protein is going through the membrane multiple times. So you can have a single protein with multiple membrane spanning domains. Most of the time, these membrane spanning domains are alpha helices, but they don't have to be. I was talking about porin proteins before, which are bacterial pores that allow water to come in so they can regulate their osmotic balance. And those are actually composed of beta-pleated sheets. All right, so there are also peripheral proteins. And peripheral proteins are linked to the plasma membrane, but they're only associated with one side of the membrane, and they're only associated with the membrane in a loose association. So they can easily be removed from the membrane. Anything that would disturb the tertiary structure of a protein would release a peripheral protein from the membrane. So changing the pH or changing the salinity, even changing the temperature slightly may release a peripheral protein. Sometimes the cell will have its peripheral proteins only transiently bound. So by phosphorylating a key amino acid, that peripheral protein could be released. Okay, and so it might have a function where only sometimes it needs to be bound to the membrane. So whereas an integral membrane protein, you would have to add detergents to a cell suspension to disturb the phospholipids to release the integral membrane proteins. Some functions of peripheral proteins might be cell cortex proteins for structural support, or things like collagen, which are in the extracellular matrix. Enzymes might be scaffolding, the cell might be scaffolding biochemical reactions, and so there might be holding an enzyme at the cell membrane. Um, Viral protein coats are good examples of peripheral membrane proteins, and some signal transduction proteins. So they may be, uh, uh, what do I want to say? They may be bound to a receptor, and then they get activated when the receptor is activated, and then they go do something else to go transduce the signal. Okay, these are some examples of peripheral membrane protein functions. Another type that we're not going to talk too much about are lipid-anchored proteins. Trimeric G proteins are a good example of lipid-anchored proteins. And these are, some people would classify these as integral membrane proteins, but they are entirely outside of the membrane. They are, most of these are, well, no, I don't want to say that. They may be cytosolic or they may be non-cytosolic, but they're covalently bound to a membrane lipid. And so they're not easily removed from the membrane because of that covalent bond. But so they don't, that you can't really classify them as a peripheral protein because they're covalently bound. But you don't really want to call them, a lot of people don't want to call them an integral protein because that implies that they're embedded in the membrane. So the CART book treats them as a separate category and calls them lipid-anchored proteins. These are covalently linked to a membrane lipid. Okay. All right. What kind of R groups would amino acids that face inside a calcium ion channel? Okay, calcium ion channel is going to be like this. It's going to look like this, where part of these alpha helices face the membrane and part of them face inside the calcium ion channel. So the calcium ions go through the channel. So this question is asking you what kind of R groups would face the inside of that channel, right? So they would have to be attractive to calcium ions. Okay, I'm going to stop the recording here before I run out of battery, and we will, I'll have another, section that picks up here.